The next thing I'm going to talk about is B cell activation. So B cell has an antibody on it. And these antibodies are like way larger than what they actually are. So in this picture, I drew them huge just so you can see them, but they're really like uptight against the surface of the B cell. And this antibody has a particular structure, and you need to know about antibody structure. So if we look at an antibody has two heavy chains two light chains, and then the top part is known as the variable region. This is the region that's randomly put together, and every single B cell has a different random assortment of variable region that can recognize a different pathogen as being foreign. Then you have the constant region at the bottom, and the constant region is the region that's going to be able to stick into the B cell membrane. There's 5 different classes of antibody, and you need to know their functions. IgM, IgD, IgG, IgE, and IgA. IgM is physically the largest. It's on the surface of the B cell membrane, and it is your initial responder. So when this B cell initially gets woken up by a pathogen or by an antigen on an antigen presenting cell or presented on the infected self cell, this is the one that's going to do the initial recognition. IgD is almost exclusively found on the B cell membrane, and it doesn't really have a specific function. So along with M, it's kind of one of the first responders, but it doesn't play a major role in B cell activation. IgG has the highest quantity in the bloodstream. So this is the one that's going to remain in the blood for the longest period of time, and it provides an aspect of memory, immunological memory. So if you have those antibodies present, that means that you've been exposed or you have a current infection with it, or at some point the B cell responded and started pumping out this antibody. So with IgG, this is the one that crosses the placenta, and after the baby's born, the baby will have some of the mother's IgG for about up to a week or so after birth. IgE plays a major role in mediating allergies and parasitic infections. We're going to talk extensively about IgE once we get to how an allergy develops a little bit later in week 11's lectures. IgA is found in tears and saliva and breast milk. It's also found in all of your mucosal surfaces. So this one lines the vagina, lines the urethra, lines the digestive tract, lines the inside of the respiratory tract. So those are the 5 major classes of antibodies. What do antibodies do? So antibodies have 2 major functions: opsonization, which is coding, and agglutination, which is clumping. So opsonization. We talked about how a pathogen makes you sick. It has to enter, it has to stick to you, and then it can start making you sick. It has to survive your immune system and so forth. So with opsonization, what opsonization does is it coats the pathogen with antibody. If the pathogen is covered with your antibodies, it can't stick to you. So that helps clear it from the body. Agglutination. Agglutination is clumping. So if we put antibody on the surface of a microorganism, antibodies are sticky. So they like to stick to not only the microorganism, but also each other. So what would typically have to happen is your white blood cell would have to eat one bacterial cell at a time. So it slowly eats one, brings it in, phagocytizes it, and eliminates the debris, and brings another one in, one by one. But if we clump them together using antibodies, you can eat like 100 bacterial cells at once. So it increases the efficiency of eating. Coding it also allows you to recognize that it's a pathogen faster. So your phagocytic cells get really excited when they see antibodies sticking to something. So they'll eat it faster and they'll eat more of them at once. So now we're going to talk about B cell activation. So the B cell has an antibody on the surface. When that antibody recognizes the antigen, whether it be on an antigen presenting cell, on the actual pathogen itself, or on that infected self cell, antibody is going to bind to antigen, and that's going to activate this B cell. This B cell is going to start dividing like crazy, and it makes many, many, many copies. Some of those copies are going to become plasma cells. Some of those copies are going to become antigen-presenting cells. Some of those copies become memory cells. And then there's other cells that they can turn into, like cells that regulate immune responses. But we're going to focus on these for this class. Now, what do plasma cells do? Plasma cells pump out large quantities of antibody so that that antibody can do its function of opsonization and agglutination. In order to pump out the correct type of antibody, we go through immunoglobulin isotype switching. Immunoglobulin is an antibody. That's why we call them IgG, IgM, Ig, so forth. Immunoglobulin for Ig. Isotype, so it's a similar type. Switching, so it switches. So we initially have IgM and D on the surface of our B cells. But once our B cells turn into plasma cells and start pumping out antibody, they're going to pump out soluble antibody in the form of G, E, or A. If it's in the blood, it's going to be G. If it's in response to a parasitic infection or an allergy, it's going to be E. If it's in a mucosal surface, it's going to be A. B cells can also become antigen-presenting cells. So that B cell, with its antibody, is going to express part of the microorganism on MHC class II. And now this B cell can go wake up other B cells and T cells to try to help fight this pathogen. The B cells can also become memory cells. Memory cells are going to stay in the body for quite a long period of time, and they're prepped and ready to produce large quantities of antibody, so that if you ever get exposed to the same exact antigen a second time, you will produce tons of antibody to clear the pathogen before you ever get sick. We have our primary and secondary immune responses, and then the concentration of antibody is graphed on this side. So during our primary immune response, it takes a really long time to produce a little bit of antibody. But the second time we're exposed, because we have these memory cells, we produce tons of antibody in a really short period of time. That will help clear the infection before we ever get sick a second time.